Yeah, sorry to cut off that song. Let's get started. I've got to finish up the last bit of speciation, and I have a lot of material to cover today on phylogenetics. But before we start, give people an opportunity to ask questions. Are there any questions? Okay, so I want to remind you where we were, give you a little bit of an overview on speciation. I talked about pre-zygotic and post-zygotic mechanisms that you can think of as like this. They're mechanisms that prevent different species from interbreeding now. So after a while, through the combined influence of genetic drift, mutation, and natural selection operating differently in different populations, you'll have divergence genetically in different populations. And the question is, if these two populations were to come back into contact later, what prevents the individuals in these two different populations from interbreeding? That is to say, what would actually cause them to be different species? And I gave you a list and described a number of mechanisms, genetic and pre-zygotic isolation, post-zygotic mechanisms such as the Johnsky-Muller mechanism, and pre-zygotic mechanisms. Then we talked about mechanisms that cause gene flow to cease initially. Right? Remember, in order for two populations to diverge, you have to, if you want to think about turn a knob, you need to turn a knob, the gene flow or migration knob to zero. You prevent individuals from migrating from one population to the other. Okay? And what type of mechanisms can do that? Well, I talked about some allopatric mechanisms. You can think about this as being the two populations are geographically separated from each other. Right? And if the nature of the barrier that's between the two populations is sufficient to prevent migration between them, well, then you can have this isolation of the two populations, and they can start to diverge one from the other. And one thing I didn't note, but so one of the mechanisms we talked about was vicariance, that is to say some physical barrier between the two populations. What might seem like a trivial barrier to you and I can actually be a substantive barrier to organisms at the right scale. So a stream, for instance, might represent a real substantial barrier to dispersal for some sorts of organisms, but we would just hop across it, right? So it really, you have to imagine there are some types of barriers that are insignificant for some types of creatures, but very significant for others. Okay. What I didn't talk about was sympatric speciation. But before I go there, I'll talk a little bit more about allopatric speciation and describe an experiment that was performed by Diane Dodd in the late 80s. Right, this is just a simple laboratory experiment. Okay, so this is all done in the laboratory. It was done with that workhorse of genetics, Drosophila lanagaster. And what she did is she grew 4 populations of flies on different media. So remember, I talked about flies before. I told you that they're kept in jars in the laboratory, and at the bottom of the jars is some sort of food source, usually a stinky food source. So usually you know it when you walk by one of these labs because they smell. Okay, and in the food source in this case were two flavors, if you will. There were starch-based food sources in some bottles, and maltose-based food source in the others. Okay, so 2 different types of food sources. I believe that usually they have starch-based food sources. Maltose is an unusual one for the flies. And she just propagated these flies generation after generation, always keeping the maltose-based flies in maltose jars and the starch-based flies in the starch jars. Okay, and after letting what I mean by when she propagates these populations, what she's really doing is taking flies from a bottle and transferring them to a fresh bottle. Right, so you take all the flies from one bottle, a starch bottle, and you transfer them into another starch-based bottle, a bottle with the starch-based food source in it. And you do that. It's a little bit repetitive, and but you do this for weeks or months at a time. Okay, what happens at the end of this experiment? After you've let this go on for several weeks, for instance, what happens? Well, the first thing she was able to do is she was able to detect differences, genetic differences in these different bottles. Remember, she's not allowing flies from one bottle to go to another bottle and mate. That's essentially she's turned off gene flow between the bottles. If you want to think of bottles and populations as being synonymous in this case. Right, so any mutations that occur in these bottles occur independently. Of course, selection is acting differently potentially in these different populations as well. So the first thing she was able to detect some genetic differences using a method that really isn't used very often anymore in laboratories, but it's a perfectly fine method for detecting differences in proteins. It's called starch gel electrophoresis. The other thing she found was that there was preferential mating among the females and males. So to say the females became choosy, and the females chose preferred males that were from the same food source as they were raised. Right, so she found evidence of female choice over time in the behaviors. This would be an example of the beginning of pre-zygotic isolation. Remember, female choice would be a type of pre-zygotic isolation for choosing some types of males, not others. And so you can see that the females at the top left, the top diagram here, the female chose a preferred males from the starch-based medium, or much more frequently, and also maltose females preferred maltose males. And then the examples with the 8 and the 9 in the cell, those are the cases where the female chose a male from the opposite food source. So the point here is that the females preferred the males that were grown on the same food source. For whatever reasons, I mean, the types of reasons, the types of genetic changes that could have been occurring here, we don't really know what they are, but there were some changes that occurred that caused the female preference. And then down here you see the control. Okay, the control is to see if the females have is it the case that they just prefer males from the same bottle, right? So the control is you take males from the same bottle and then you mate them with males, or see if they prefer males or not prefer males from another starch bottle with the females from a starch bottle, right? That was the control she did. And the point here is it's not just starchiness, it's not just the food source that matters, it's the fact, it's not just the bottle that matters, rather it's the food source that matters. This is a fairly famous experiment showing that you can actually get pretty rapid divergence in some of these reproductive isolating mechanisms. That's the important point here. Now, I want to talk about sympatric speciation last. In contrast it with allopatric speciation. Allopatric speciation, the populations are physically separated from each other. In sympatric speciation, you have speciation occurring even though the populations might overlap. Right, so the point here is that the males and females from the different populations are coming into contact in principle all the time because they live in the same place. So how can you actually turn off gene flow? How can you prevent mating between the two populations? Okay, and there's a number of different ways you can do this. It could be that, for instance, that you have temporal, somehow that, for instance, the time at which mating occurs in these populations has changed very rapidly. But there's actually a pretty good example that's pretty easy to understand, which I'll describe, which involves that particular fly. It's a pest now. It's native to North America, and ancestrally, that is to say, in the distant past, like 100, 150 years ago, the life cycle revolved around the hawthorn fruit. So hawthorn fruit are pretty they're common in the northeast, and there's people actually pick these berries and eat them. Right? They're called hawberries. They have a hawthorn fruit, and as I described last time, the males buzz around these. There's a male buzz around these hawthorn berries, hawthorn fruit, and they mate with a female that comes to the hawthorn fruit, and then the female oviposits eggs on the hawthorn fruit. The eggs hatch into maggots. The maggots spoil the fruit, and then they fly. Then they go off and do their thing. But the point is that there's some fidelity of the Ragolitas insect to the actual hawthorn fruit. As I say, if you hatch on a hawthorn fruit, you're going to come back and mate around the hawthorn fruit, and also lay your eggs on the hawthorn fruit if you're a female. That's their life cycle. It's very exciting. Well, that's the hawthorn. Well, about 100 or more than 100 years ago, some of these Ragolitas insects became, if you want to say confused, but for whatever reasons, they started to lay eggs on, say, apples or cherries. Right? This is when they first became really noticed by people because the people whose orchards they were laying their eggs on noticed that a lot of their fruit was all of a sudden spoiled. Right? So this is something that all of a sudden people became really aware of these stupid insects that are spoiling our fruit. So here you have an apple, and you have males who happen to hatch on an apple, buzzing around the apple, mating with females that come to the apple who also hatched off the apple fruit, and oviposit their eggs on the apple fruit. Okay? Now the interesting thing is because these flies have this host fidelity, they have some fidelity to the type of fruit they were hatched off of, you now have an isolation. You've turned off gene flow between the apple-laying Rhagoletus and the hawthorn berry-laying Rhagoletus, right? The hawthorn fruit and the apple orchards could be in the same geographic area. These two different, the hawthorn-loving flies and the apple-loving flies can buzz right by each other, right? They might wave to each other as they're going on their merry way today, but they only mate with individuals that prefer the same fruit. Okay, so that's the point here. So this is an example of sympatric speciation. Now, it's not clear if it's speciation yet, but it's at least an example of a case where two populations living in sympatry do not exchange genes, right? Where you've had gene flow eliminated. So there you go. That's a bit of this. So what's happened over the last 100 years? Well, like I said, it's now a major pest. That's why it's interests a lot of people. You have what's called, people call this type of mating preferences assortative mating. As I say, hawthorn males with hawthorn females, apple males with apple females. And over the last 100 years, once again, you've had rapid divergence in these two different populations. Okay? Once again, natural selection, mutation, and genetic drift are working independently in these two different populations. So you can have different alleles becoming fixed in the two populations. They can start to differentiate from each other. Okay? And once again, they used a fairly older technique to detect these differences, but it doesn't matter. It's a perfectly reliable method. And they've also discovered other differences in these different populations. So for instance, the hawthorn agates, the ancestral developmental time is about 60 days. These apples, the Rhagoletus that lay eggs on apples actually develop faster. Right? So some of these changes have caused changes in things like the development of the developmental time. Okay, so what I want to do is go to my next lecture. That was all I wanted to say about speciation. Okay? This lecture is going to be about phylogenetic analysis. And phylogenetic analysis is about the tree of life, right? It's about how we infer how different species are related one to another. This is actually my area of expertise, I guess I would say. And remarkably, this has been an area that maybe 20 years ago, very few people were interested in. This subject I was a graduate student, happened to be interested in for whatever reasons. And now, with the rise of bioinformatics and computational biology, many people are interested in phylogenetics. It's one of the very first things people do with DNA sequences when they come spinning out of these automated sequencers is they make phylogenies. This is about the second thing they do is after they align the sequences. So it's one of these phylogenies are made all the time. This is kind of an outline of what I want to talk about. I want to talk about trees, discuss some of the aspects of trees, such as rooted, unrooted, how you can actually root trees. I want to talk about how you estimate phylogenies, to say how do we decide which is the best representation for the tree of life, and then give you some neat examples of phylogenies over the last 10, 20 years. So it turns out, phylogenies, this metaphor that there's an underlying tree of life that connects all organisms on Earth is an old one. It even predates Darwin. But Darwin gave a lot of evidence for the idea, and in fact, it's such an important part of the origin of species that the only figure in the origin of species is this one on the right. That's the only figure in the origin of species in which he's just trying to depict an imaginary group of organisms and how they might be related to one another. And then in his notes, this is some doodle by Darwin in his scientific notes. This is an example of a phylogenetic tree. So he was thinking explicitly about trees as well. Stop doing that. Immediately after the publication of the origin of species, there was a lot of interest in phylogeny. Since the origin of species, there are people who took the view that, well, if there is an underlying tree relating to all species, then what is that relationship? And so this is an early tree by Haeckel in the early 1860s. Haeckel was a German biologist who was an early proponent of Darwin's ideas. But he made this tree. It's not clear how he actually decided upon the relationships, but there's a couple of remarkable things about it. One is that it's fairly accurate. There's aspects of this tree that are pretty good. So for instance, it's all in German. So if you go to the very top, you have mentioned, that's humans. And then he has the orang, the gorilla, the chimpanzee, and the gibbon as all our closest relatives. It doesn't have the exact relationship, right? But he realized that those were our closest relatives. And then below that, he has monkeys and so forth. So that's the first thing. And then, of course, the other thing is it's just really kind of cool, pretty tree. You put bark on it, it looks like a tree, right? Which people don't have the time to do those things anymore. All right. So let me put the screen up for a few minutes while I talk about some of the basics of trees that you probably need to know, or you do need to know because you want to do well on the test. So the first thing is that you need to realize that this is an area that's highly computational, right? And it'll become obvious why that's the case. People do not make trees by hand anymore like they did in Haeckel's day. They do it on the computer. And in the computer, trees don't have a direction, right? So the tree of life obviously has a direction. That is to say, time goes in one direction. There's a bottom of the tree, what we call a root of the tree, the continuous metaphor with trees, and it goes forward in time until you get to the very tips. The tips would be the species that are alive today. But in computer memory, these trees are not rooted. So this is an example of an unrooted tree. I will label it with species names, which I'm just going to call A, B, C, D, and E. Okay, that is an unrooted tree. As an aside, these points where these lines come together, these are called nodes, and the lines are called branches. There's actually a mathematical terminology for these two, but all biologists call them nodes and branches. If you're a mathematician, you call them vertices and edges, but you don't have to know that. These tips are often species names, but they could even be just individual DNA sequences if you're talking about sampling individuals from a population. Okay, so we're going to be thinking of these as individual species. This would be species A, species B, species C, species D, and so forth. So that is an example of an unrooted tree. Now, the idea is that you can actually convert an unrooted tree into a rooted tree by choosing one of the branches. In this case, there's 7 branches, 1, 2, 3, 4, 5, 6, 7, and choosing to root the tree along that branch. What that would mean is you have to break that branch, put a kink in it, and pull the entire tree down by that point. So we could root the place anywhere, the tree anywhere. Let's root it here. What type of tree results? Well, if we root the tree here, we're going to have the X is going to be down here, and there's going to be a kink in that branch right here. Right? We kink this branch, I just broke it in two. Right? Off to one side, doesn't matter which side, we put D and E. And then on the other side, we have A, B, and C. But notice that the first branch here, the first branching goes to A, B, and C. So we'll say C on one side, and then A and B on the other. This is an example of a rooted tree. Rooted. That is the root. Okay, that is the root of the tree. That would be an example of converting an unrooted tree into a rooted tree. Just pick a branch and you kink it. All right, is that clear? A couple of little things that some people find confusing. You can rotate these branches and not change the meaning of the tree. So for instance, I could put A here and B here, and that is the same unrooted tree. I've not changed the meaning. So you can rotate. If you can imagine these were lichen logs, or not lichen logs. What are the ones with the little sprockets with the, what's that? Tinker toys, maybe. I don't know. But you know the little toys where they have the little dowels and you have the little places. Just imagine you have a dowel each one of these and you can rotate it, all right? I can't remember the name. I used to have these toys. The tree topology does not change based on rotation, all right? Yeah, brain fart there. Okay. So in practice, how do people decide what branch to root it on? Well, there's a number of different ways of doing this, but the method you will hear about in class, in labs, for instance, and also the most frequently used method by evolutionary biologists is called the outgroup method. And the idea with the outgroup method is that when you do your analysis, presumably you're interested in the relationships, as an evolutionary biologist, presumably be interested in the relationship to some group of organisms, your favorite group perhaps. I mean, pick a group, maybe you're interested in some group of lizards, maybe in moles, right? So what you do is you put the species in your analysis that you're interested in, your different lizards, right? And then you choose one species of lizard that you're absolutely certain was not inside that group. So for instance, if you're interested in phylogeny of lizards, something that is not a lizard is a dog, right? That would be an example of an outgroup species. The dog would be the outgroup. You throw the dog DNA sequences, whatever characteristics you have for the dog, and compare them to the lizard sequences. And what you'd have is you'd have your lizard sequence, a tree, this unrooted tree would have your 4 lizard species, and then you have your dog, say, right? And the idea is if this is your outgroup sequence, this is your outgroup species, then the branch you decide to kink when you root it is this one, the branch leading to that outgroup. That's all there is to it. Now, as an aside, dog, if you're interested in the phylogeny of lizards, a dog would be a bad outgroup. I mean, anything that's not a lizard would be a potential outgroup, but in general, you want an outgroup species that's as closely related to the species of interest, the group of species of interest as possible, but it's not one of them. So the best one would be to choose maybe a crocodile, which is more closely related to lizards than a dog is, for instance, right? Or if you're interested in just a small group of lizards, you might pick another lizard that just doesn't happen to be in that group. Okay? Anyway, so that would be an example of using, this is one method for converting unrooted trees to rooted trees because the computer programs spit out unrooted trees, almost always. That's what the computer programs spit out. Right, so I wanted to talk about, so I talked about trees, rooted versus unrooted, and rooting them. I want to talk a little bit about the combinatorics of trees. Does anybody know what the word combinatorics means? Yeah, go ahead. No, it doesn't. It can be, maybe because I don't know enough about lizards. Let's say you're interested in the phylogeny of apes, right? So gibbons, orangutans, gorillas, chimps, and humans, right? And so if there's 5 species, you could collect DNA sequences from all those 5 species and compare them, and the computer program would spit out a tree like this with 5 species. If you wanted to root that tree, what you have to do is you have to add at least 1 more species, right? And so you want to pick something that was not an ape, right, to throw into your analysis so that you have a tree with, instead of 5 species, 6 species in it, right? The 5 species of interest plus the outgroup. So as an example, now dog would be a good example, right? Dog is not an ape. So you can take your, I don't know what type of dog you have, take a little blood in a nice way and sequence the DNA from your pup and throw it into the analysis, right? Your dog now is acting as the outgroup species. There are better outgroup species we could probably choose. So for instance, you could pick another primate that you're absolutely certain is not a ape. So for instance, would be a good one. A baboon would be good. It's an old world monkey, fairly closely related to apes, but definitely not an ape, right? So that would be a better outgroup species because the amount of time these trees, you can make mistakes estimating these trees, which we'll get to. The longer the branch of the outgroup, the less certain your rooting is. Okay. Anyways, I want to talk about combinatorics. Anybody have an idea what combinatorics is? All right, it's the mathematical field of counting things, all right? So that's how I describe it. I don't think a mathematician might shoot me, but it's counting things. So how many possible trees are there out there? If you have a problem with 4 species, say, what is the realm of possibilities? How could these species be related? So let's go through an example with 4 species, and I'll show you the possible relationships. We have this tree. That's one of the possible unrooted trees for 4 species. Here's another tree. And there's one more. There's only 3 possible unrooted trees that can explain the relationships of 4 species. If you're interested in how you actually derive that, come to my office hours. Otherwise, it's probably not worth talking about right here. But that's all the possibilities. There's only 3 ways that the species can be related to each other on an unrooted phylogeny. So if we wanted to make a graph, or a table rather, what we'd do is maybe we'd have n, the number of species in your analysis, and here would be the number of unrooted trees. All right, so what I just said is if you have 4 species, there's 3 possibilities. Okay? Now, if you have 5 species, I'm not going to draw these out. There's 15 possible trees that can relate them. Okay? If you want to try and figure it out, you can do it. But if you get to 16, you've made a mistake, right? You've overcounted a tree. If you have 6 species, there's 105 possibilities. I think 7 is 945. 8 is 10,395 possible trees. When you get to 9 species, the number of possibilities is 135,135. 10 is 2,027,025. Notice what's happening. The number's getting big fast, right? By the time we get to 20 species, I don't know the exact number for 20. By the time we get to 20 species, we'd be talking about roughly a mole of trees. Avogadro's number of possible trees, okay, which is a lot of trees. That's just like 20, 20, which is a handful of trees. By the time you get to 50 or 60 species in your phylogeny, you're talking about more possible trees than there are atoms in the known universe. And I don't know how people count the number of atoms in the known universe, but you're talking about roughly 10 to the 60, right? And you exceed that number in the number of possible trees when you're about around 50 or 60 sequences, right? Trees of 50 or 60 tips to them, right? So the problem is this, is that if you're interested in finding the best tree, the first problem we're faced with is what they call this combinatorial explosion in the number of possible trees. If you're interested in 20 species, you have roughly a mole of trees to consider, only one of which can represent the true relationships of the species. Right? So how do you choose among all these different possible trees? Well, I'm not going to recommend doing this, but if you had a small number of trees, you just pick one of these 3 trees at random, right? And you get the right tree 1/3 of the time. Would you agree with me on that? It would be the stupidest method in the world, but you could pick one of the trees at random with 4 species and get the right tree 1/3 of the time. But when you get to 10 species, that same method really stinks, right? It wasn't very good for 4 species, but with 10 species, it's really bad because you only have 1 in 2 million chance of getting the right tree. You might as well play the lotto, right? So we need to have a method that somehow increases our chances of getting the right tree. And also, it should be based on evidence, right? We shouldn't just be picking things out of thin air. That's not science. That's just lame, right? So let me show you, I'm going to illustrate how people actually estimate phylogeny. One of the frustrating things about a course like this where I'm teaching you all about phylogeny in less than 50 minutes is that the field is actually quite complex. And I'm going to be describing one method for inferring trees, but you should realize that there's actually what I consider better methods out there, okay? But I'm biased because I invented some of those methods. But so you can't take my word for it at face value, so I'll just tell you that right now. But this is an example of what we're going to be basing our trees on. This is what's called an alignment of DNA sequences. The idea is that for each of the 4 species, I labeled them here 1, 2, 3, 4 instead of A, B, C, and D. The idea is that for each of the 4 species, you went out and you sequenced a particular gene, okay? And you compare the DNA sequences for that gene across all 4 species. And so there's computer programs that do the fine shuffling to make certain you have the columns, that each column is a different position in the sequence. There's computer programs called alignment programs that will perform this step for you, okay?